my presentation is about biomarker and prospect for interventional trial at the pre-ataxia stage in different spinal cerebellar ataxias. And I would like to emphasize on three important topics. Number one, the general overview of spinal cerebellar ataxias. The second point will be about preclinical state in SCA2. And at the end, I'm going to speak about the biomarkers in SCA and in particular case of spinal cerebellar ataxia type 2. As you know, the hereditary ataxias are an heterogeneous group of neurodegenerative disorders caused by the degeneration of cerebellum and its afferent and efferent connection, the spinal cords, peripheral nerve, and the brain stem. The most common of these spinal cerebellar ataxias, the most common of these spinal cerebellar ataxias are the polyglutamine expansion disease. And the worldwide prevalence of SCA is about 3 to 5 cases per 100,000 inhabitants. And the clinical picture is characterized by cerebellar disorders associated with other neurological manifestations, such as peripheral neuropathy, corticospinal signs, and cognitive decline, etc. Okay, in this map, you can observe the frequency distribution of the different molecular subtypes of spinal cerebellar ataxia worldwide. Sorry. Okay, perfect. The worldwide prevalence of spinal cerebellar ataxia, as you know, is between 3 to 5 cases per 100,000 inhabitants. Latin America is one of the most affected area of worldwide, and there are some countries with founder populations, such as SCA 7 in Mexico and Venezuela, SCA 3 in the South of America, and SCA 2 in Cuba, where the frequency is about 90% in comparison with the other molecular subtypes. You can see here the distribution of neuronal loss of the brain of patients with different molecular subtypes of ataxia. As you can see, the more severe degeneration in SCA2, SCA3, and SCA1. However, in SCA7 include a severe retinal degeneration, and in SCA6 and 8, the neuronal loss is commonly less severe degeneration. Now, different studies have been carried out in preclinical subjects to identify the most important clinical feature during evolutive stage. And in these cases, the most frequent clinical alteration include the mild incoordination in SCA1 and SCA2, gaze nystagmus in SCA3, painful muscle cramp in SCA1 and 2, and hyperreflexia in SCA7. Okay, the main different studies have been carried out in relation to the preclinical electrophysiological evaluation in different SCA, and the most or the main electrophysiological alteration include corticospinal tract dysfunction in SCA1, REM sleep behavior disorder in SCA3, and ocular movement disorder in SCA6 and SCA7. Now I'm going to move to the second topic, which is about the preclinical state in SCA2 and other polyglutamine expansion disorder. As you know, the SCA2 is the second most common spinal cerebellar ataxia worldwide. In Cuba, we carried out an epidemiological survey in the whole country, and we identified more than 100 patients with different hereditary ataxia and around 10,000 at-risk descendants belonging to around 213 unrelated family. The national prevalence rate is about 36 mutation carrier per 100,000 inhabitants. As you know, the province with higher prevalence rate is Holguín, with about 48 patients per 100,000 inhabitants and around 200 SC2 mutation carrier per 100,000 inhabitants. In Cuba, every year, 45 new SC2 cases and certified preclinical subjects are diagnosed. Moreover, 20 patients die due to the disease every year. Let's see here the epidemiological feature of SCA2. So, global epidemiological data show that SCA2 has a wide geographical distribution across the world, being the second most common subtype of autosomal cerebellar ataxia worldwide after only to SCA3. In this map, you can see the large population of SCA2, and it is located, for example, in Mexico, Venezuela, South Africa, India, Italy, and in Cuba, where the frequency, the relative frequency is about or more than 10% in comparison with other molecular subtypes of ataxia. Concerning the clinical picture of SCA2 patient, I would like to say that the most important alteration include the progressive cerebellar disorders in 100% of patients, slowing of saccade is moving in around 98%, peripheral neuropathy, signs of motor neuron involvement, autonomic abnormalities, sleep disturbances, and cognitive disorders. Now, I'm going to show you the natural history of SCA2. It can divide it into important or in 3 important states and 2 states before ataxia onset. The asymptomatic state or phase is defined by absence of signs or symptoms of the disease, including clinical, electrophysiological, or imaging evaluation. The programmal or preclinical state is characterized by different functional anatomical signs of neurodegeneration. No motor symptoms or mild clinical motor signs in the absence of cerebellar syndrome. You can see the first alteration is the saccade slowing that appears about 15 years before ataxia onset. Also, we can identify the postural instability that appear around 10 years before ataxia onset, and it is identified by the use of the posturography. Okay, close to ataxia onset, we can identify the abnormal tandem gait and coracospinal tract dysfunction. Concerning the most important preclinical feature in spinocerebellar ataxia, I would like to emphasize in the painful muscle cramps that appears in about 81%, sensory abnormality, mild cerebellar manifestation, also hyperreflexia, autonomic dysfunction, olfactory alteration, and cognitive decline. Concerning the most common imaging alteration or criteria, we identify the mild atrophy of the cerebellar and brain stem in preclinical subjects. Concerning the electrophysiological criteria, I would like to emphasize in the slowing of saccade its movement, the increase of anti-saccade error rates, the prolongation of anti-saccade latency, alteration of the REM sleep, and increase of the latency of the P40 component and the central motor conduction time, and finally the decrease of sensory action potential according to the sensory nerve conduction studies. The paraclinical neurological criteria are very important in this moment, and the most important includes the alteration of the gait variability, okay, and the alteration of the sway postural control. Okay, now I'm going to talk about the final point, which is about the biomarker in spinocerebellar ataxia and in SCA2 in particular. I would like to start by talking about the definition of biomarkers. Biomarkers are objective measures that reflects the neurodegeneration, provide insights into disease physiopathology, and evaluate the pharmacological responses in clinical trial during preclinical or manifest state. They are very important because they improve clinical diagnosis, state disease course, monitor disease progression, provide physiopathological clues, assess the efficacy of clinical trial, and it is very important to the selection of patients for clinical trial. We classify the biomarkers in two important groups. The first group includes the biomarker related with the polyglutamine toxicity of genetic damage. And the second group is relation to the progression of the preclinical state. Concerning the polyglutamine toxicity biomarker, I would like to emphasize this includes the mild cerebellar feature, saccade slowing, pyramidal sign, and cerebellar structure loss according to the imaging studies. Now we are going to show you the imaging biomarker. Observe here, the volumetric data reveal the degeneration of cerebellum in brainstem, in particular of the pons in SCA2 preclinical subjects when we compare with the control group. And in the other graph, you can observe that the pontocerebellar atrophy is associated with a CG repeat and predicted age at onset in preclinical SCA2 individual. The electrophysiological biomarker related to the polyglutamine toxicity includes the saccade, the maximal saccade velocity, and you can observe the saccade velocity is significantly reduced in preclinical SCA2 subject, and it is depend on the CG repeat 2. Preclinical subject with a large CG repeat expansion size had stronger degrees of saccade velocity. Finally, of this part, I want to show you the postural instability in SCA2 preclinical study. You can observe in this graph the preclinical carriers show an increase of oscillation frequencies for fit together and tandem position tests. And the higher oscillation are close related with the large CG repeat. It is a very, very important parameter or paraclinical parameter. In relation to the biomarker of progression, I am going to show you the most important includes the cognitive decline, mild cerebellar feature, cognitive alteration according to the electrophysiological evaluation, sleep disturbances, and somatosensory deficit, saccade slowing, and pyramidal sign. They are different objective measures that you can see in this chart. Okay, the swim time discloses an increase of variability in preclinical subject when we compare with the control group. And the variability of swim time in the preclinical subject is significantly correlated with the time to ataxia onset during walking, natural walking. It is a very, very important paraclinical biomarker that is for the use in clinical trial and to evaluate the progression of the neurodegeneration during preclinical state. The anti-saccade abnormalities have increased in preclinical subject in relation to, for example, the inhibition anti-saccade error rates and in comparison with a healthy control. Likewise, the latency of correct anti-saccade is increased in preclinical subject when we compare with the control subject. But latency are inversely correlated to time for ataxia onset. Okay, the next one. Finally, I'm going to develop a summary, the non-motor and motor alteration, brain imaging, pathology, and electrophysiological abnormality define the preclinical stage in polyglutamine expansion disease. The preclinical biomarker description in Cuban SCA2 gives novel opportunity to start the clinical intervention before ataxia onset, and it represents a model to extend to other spinocerebellar ataxias. The experience acquired by the Cuban ataxia group is a strength for the development of an international collaborative project and scientific networks studying hereditary ataxias. Okay, now I show you the Cuban multidisciplinary network of hereditary ataxias located in different parts of our country, with the objective to develop many important epidemiological and medical assistance to our patients across our world. Thank you very much for the nice attention.
Okay, thank you, Luis, for this talk. I would like to repeat again that I'm really happy that you could participate here and that everything worked so well. This is great work that you're doing with the SCA2 in Cuba. Maybe to save some time, could you stop your screen sharing, and the next speaker, Jeremy, could already start to share his screen to save some time. I've seen one question that came from Massimo, and he was interested in the loss of olfaction, and the question was whether there is an association between the Parkinson phenotype and in SCA2 and the loss of olfaction, or whether this is something happening in this with the same frequency and also in ataxic SCA2 patients.
Okay, concerning in relation to the Parkinson phenotype in SCA2, we have some cases with this phenotype presentation, and in these cases we evaluated. We carried out clinical and in some cases imaging evaluation, but a structure evaluation. And the most important alteration in these cases is the coexistence of the cerebellar disorder and other Parkinson's alteration. And concerning the imaging evaluation, we identify mainly the alteration in the cerebellar and brainstem atrophy, and in some cases the striatum alteration. We are not seeing the evaluation between the loss of the alteration in the SCA2 and Parkinson's in relation to other studies.
Okay, thank you. Unfortunately, we have to move on to the next due to the lack of time. And the next, thank you again, Luis. And next speaker will be Jeremy Schmahmann from Boston. And he has been a pioneer in developing scales and outcome measures. And he will today report on a new outcome measure, which is a patient-reported outcome measure, which may be extremely important for future studies. Hi, Jeremy. Good morning in Boston, and go ahead.
Good morning, Thomas. Good morning, everybody. Good afternoon, everybody. It's my great pleasure to be here. Thank you for the invitation. Wish I could see you all in person, but this is what we have, so here we go. The question is this measure ready for trials? And what I'd like to do today is actually take you through the study, and you can make the decision yourself at the end of that. This is a collaborative work. We had a student, Samantha Pierce, who's now in medical school, Jason Mackmore, who's my lab manager, and involved deeply in the work we're doing here. And Gil, the Italian at Biohaven, has been very helpful throughout the project in statistical analysis and in guiding us along the road to follow the methodology of the development of a patient-reported outcome measure. The background of this is that the assessment of ataxia, as you know, has largely been confined to clinical rating scales with instrumented quantitative approaches and the imaging biomarkers. These are administered by us. They're agnostic to patient experience and the subjective assessment of the response to intervention. We know from other diseases that the patient-reported outcome measures are in fact important as determinants of disease severity, evolution, and response to intervention, and there is no ataxia-specific PROM. So the plan was to go ahead and actually develop one and validate it from ataxia. We did this by following the guidelines of the patient-reported outcome measures information system and also the FDA guidelines, and there are 4 distinct phases. The first was the conceptual framework, which is to generate the issues that are relevant to patients and then divide these into domains. And here's what we did. We gave a semi-structured anonymous survey, which was the 6 questions below, to patients with ataxia. This is from the National Ataxia Foundation annual conference in 2018. Now, the people who show up at the NAF conference are all the SCAs, Friedreich's ataxia, other recessive ataxias, episodic ataxias, the recessive ones, multiple system atrophy, sporadics, gluten, and then people who don't know what they have. So it's a whole range of ataxias. And there were 6 questions that were given to people in a very open-ended format. Tell us everything that you have. What are all your symptoms? What activities have been involved by your disease? What hobbies or activities have been changed? What has not been affected by your disease? Have your thinking or your emotions changed? What do your doctors not ask you about that you want us to know about? You tell us everything you can. So we had thousands and thousands of comments from people, everybody giving us all their symptoms. And then we went through this, and then informed by my own input and review of the literature, we took these items and categorized them into unique domains or subdomains based on their symptoms and on their activities of daily living. And that was used to develop a preliminary version of the PROM, and the severity and the frequency was scored on a Likert scale from 0 to 4. We then took this initial version of the PROM ataxia, and we went on to phase 2, which is a cognitive debrief. And when we asked people to actually come in when people could come in before COVID. And we had focus groups based on Thomas's severity staging of normal gait, impaired gait but independent, walking aid required or wheelchair. And then these focus groups of these 17 patients were asked to assess the content validity, the readability, and the comprehensibility. Did the questions make sense? Were they important to you? Were they relevant? Was there something we should change about the question? And we applied metrics for responsiveness to severity level, the internal consistency of the items themselves, and then the acceptability and reliability of the data that came out of the initial evaluation. And then we incorporated the feedback from these focus groups into the final PROM. We changed wording, we left out some questions, we added in some questions based on the feedback from the patients. That's the cognitive debrief. That gave us a PROM ataxia, which I presented to the National Ataxia Foundation investigators meeting a year ago, and got some wonderful feedback that had to do with what's the test retest reliability. So we went ahead and did that, and we sent the PROM out to people who the National Ataxia Foundation identified for us. This was completely anonymized, and so this was important for the IRB approval. All we had was the diagnosis and severity level, stages 1, 2, or 3. And people were asked to complete this 3 times on day 1, day 15, and day 30. And then this, because we had 78 mostly novel ataxia patients, but because they were anonymized to us, there could have been some overlap. We used this data to re-explore some of the correlations that we had from the initial data set. And then finally, we did an external validation in 20 ataxia patients who came into the clinic, and we evaluated the PROM in relation to established measures of motor ataxia, emotional well-being, and quality of life. Now, here are the results of the study. The first, the conceptual framework and domain selection and item pool development. There were 147 people who answered the call to tell us their symptoms from the National Ataxia Foundation meeting in 2018. They broke down into stages 0 through 3, as you can see. About 7% were walking fine, 51% were impaired in their gait, 37% required some kind of a assisted device, and about 5% were in wheelchairs. So we had 3,855 items that were presented in these categories of all the symptoms they had: activities, hobbies, what was spared, thinking, emotion, and other relevant troubles, breaking down into different items reported for each of these. And then what we did was we took these 3,800 items and we put them into different domains of function. We broke this down, which you'll see in a moment. And then we conceptualized these into 3 overarching domains and came up with a total of 70 items. So here are the 3 domains: the physical domain, the mental health domain, and the activities of daily living domain. The physical domain, this comes from patients now. This is all patient-derived. It's a really important concept to keep right up at the top of our thinking here. Speech, vision, neuropathy, dizziness, muscle control, fatigue, bulbar symptoms, balance coordination, sexual ability, and sleep. The activities were ADLs, what you can do on your own and what you need to have instrumental activities to help you with. And then there was emotional dysfunction and social health. So it was sort of cognitive and emotional here. In terms of the other aspects of this, the content, validity, readability, and interpretability, we had 17 people in focus groups who gave us some feedback on the initial prom. They all thought that it was very understandable. The questions were great. They were all important, and by and large, they were relevant to them. Depending on the severity of disease, relevance would be more or less important. The responsiveness to gait, remember this is the relationship here is to the motor severity, just in terms of the 4 issues of independent walking, and fine, independent but not but impaired, requires some kind of a walking assistant device or in a wheelchair. The responsiveness is actually very strong. So this is for the physical domain, a chi-squared of 0.45. This is the ADL domain of 0.57. And then the mental domain, most emotional and cognitive, correlates, but not so strongly as we would probably expect. Now, the internal consistency and reliability were high with 0.9s for the physical and the ADL and 0.88 for the mental domain. The data was acceptable and reliable in terms of the mean and the median scores. And there was a floor ceiling effect that had the full range of severity across most of the items. So it seemed like we had a successful scale based on the 17 participants. We then went ahead and performed the retest reliability. And here we had 78 people who filled this out 3 times, day 1, day 15, and day 30. And what we found here is that there is a very good correlation between the first 2 weeks and the second 2 weeks. In the physical domain, the total and sections 1 and 2, which I'll show you in a moment, the activities of daily living domain, very strong correlation. The mental domain was similar, maybe not quite as strong as the first 2, but this shows that the retest reliability, when the disease has not changed within that period of time, is very strong. So it would be reasonable to use it for assessment of change over time, we think, although we haven't shown that yet, and I'll come back to that later. We re-explored the responsiveness because we now had 78 subjects and not only the ones that we had in the patient focus groups. And here we see again the physical domain and the ADL domain are correlating nicely with the gross division of the motor impairment. Mental domain, the correlation not so strong. Now, here we went into phase 4, which was to look at the PROM against other established measures of motor control, quality of life, and mental cognitive function. So what I've done is the scores here are coded by color, where green is a correlation of greater than 0.6, yellow is 0.4 to 0.59, and red is less than 0.4. So good to less good correlation. So the PROM physical, how people are doing with sections 1 and 2, but 1 and 2 is severity and frequency of problems. correlates very well with the FARS, one which is a functional staging, the activity of the daily living scale, the NeuroQoL for the upper extremity, the lower extremity, and the fatigue scale, and also with the Biohaven in-house version of the patient impression of function and activity of the daily living scale, which derived from the similar kind of questions that we have discussed with Gil. There is a very good correlation with the PROM-ADL as well, with bars, the brief ataxia rating scale, which is our version, a shorter version of ataxia assessment, as well as the FARS 1 and 2, and the arm, leg, and fatigue, and with the PIFAS. The SARA and the bars have similar correlation, both with the PROM physical and the PROM ADL. We did both bars and the F. This is the F SARA, which is a 4-item functional SARA that the FDA had mandated that Biohaven use in their clinical trial. Now, the mental function here has the sort of relatively, there's modest correlations in the yellow with the Beck anxiety inventory, the Beck depression inventory, and less so with the FARS, but there is some with fatigue and with the PIFAS as well. So we have, I think, relatively strong correlations between the PROM ataxia and those measures that people report about their own life experience through these different scales. And there's a modest correlation between the PROM ataxia and the motor scales, the bars and the F-SARA. But you'll see as we look at the scale items in a moment that this is actually understandable. So here are the items of the PROM ataxia. And I'll show you in a moment why some are outlined in red. But this includes, I feel unsteady on my feet, trouble walking. I feel as though I may lose my balance. I do lose my balance. I'm clumsy. I find myself stumbling. I have trouble controlling my arms and legs. My hands shake. They shake when I'm carrying things. I have numbness and tingling, muscle stiffness. My speech is slurred. This is from never to often, a Likert scale. People have trouble understanding what I'm saying, trouble swallowing, difficulty drinking, blurred vision, depth perception, bladder, bowels, falling asleep, sleeping through the night, fatigue, dizziness, loss of sex drive, sexual activity. Again, this is from patient their own reports. Then section 2 is not, this is never to always. This is no difficulty, can't do. It's a severity scale. Carrying things, walking without assistance, catching myself, sport, bending down, picking up things, getting off the floor, rolling in bed, perform tasks with my hands, using my hands, writing, typing, doing my job, doing household work, yard work, shopping, caring for myself, getting out of a car, public transport, cooking, cutting food, utensils, dressing, moving about the house, showering, brushing my teeth, shaving, getting out of bed, off the toilet, going to the bathroom, feelings of being sad, down, depressed. This is now the mental thing. Irritable, losing activities and interest in activities, trouble controlling my emotions, anxiety, worry, and stress, less motivation, isolated, impacting my social interaction with family and friends, becoming anxious, feeling self-conscious, thinking of words I want to say, remembering conversations, multitasking, learning new skills, making decisions, comprehending things, and I can follow directions well. So these are the 70 items, 70 questions reduced from these 3,500 things people told us about that has gone through these iterative stages that patients tell us are important. They understand the questions and relevant and correlate well with the other aspects of assessment that I've mentioned to you. Now, there's 70 questions there. It is long. Nobody complained about the length of it, but the question here is, can you make a shorter form of this that would be just as good? So what we did here was we adopted the same approach that we did in developing the brief ataxia rating scale from the ICARS, the International Cooperative Ataxia Rating Scale. And we rank ordered the different items from 1 to 70 of the scale into those that correlated well, intermediate, or poorly with the rest of the items. So the 69 items correlating with this one item, that was the relationship. And we found that there were some that were the heavy hitters in the green. Now, what we did was then you can either do a non-a priori or an a priori approach. The non-a priori, you just take the 10 items that are strongest correlating with the remaining 69 items. Or, as you did with bars, you have an a priori approach that you want to know what's happening in each of these sections, because this is what the patients told us. The patients told us these sections count. So we did physical section 1 and 2, which is frequency and severity, ADLs, and then mental item 1 and 2, which is cognition and emotion. And here's what we have is the primary ataxia short form with a priori has this very strong correlation with the remaining 60 items. So these 10 items, the short form correlation with the 60 items is very strong. So it certainly makes sense that you could use just those 10 questions, but there's a lot that you leave out as a consequence of that. That becomes a separate issue. So here are the 10 questions that were the heavy hitters in terms of the correlation with the other items. I think I may lose my balance. I do lose my balance. I have trouble controlling my arms and legs. I can walk without assistance or not. I can catch myself when I stumble. I can do things with my hands, household work, cutting up stuff. It impacts my social interactions, and I can multitask or not. So these things fail as the disease evolves, at least in terms of the motor phenomenology. So where this takes us to is if now I have this primary ataxia with 70 item, it's a 3-domain, 70-item questionnaire, which comes from the patients. And we developed this in 4 phases. We generated the items, we did a cognitive debrief, a retest reliability, and we validated that against other measures. And then we developed a 10-item short form. I think what we conclude is that this is the first cerebellar ataxia-specific patient-reported outcome measure. It has excellent retest reliability, and it is validated. We think it has the potential to empower the ataxia community to improve patient care and serve as a biomarker in trials and help facilitate development of new ataxia treatments. And we'll have to see whether it is responsive to change over time and whether the primary ataxia, in fact, is useful. Now, companies may find that the short form is all they need, but we're going to lose data for the long form. So that's going to be a discussion as to what your need is. And the plan is to look at the change over time, responsivity of the problem to change over time over a 3-year period. And I'll end there with a couple minutes to spare. Thank you for some of the students who helped us early on, and Melissa Bynum from Biohaven has been a real supporter of the whole project, supported by NAF, Biohaven, our own fundraising group, and the group here at the Ataxia Center who's been helpful in making all this happen. And of course, we're grateful to our patients and to NAF who have allowed this to get to the stage that it has, and working together, recognizing patients as our collaborators and experts by experience, allowing us to get to this point. And for those who want to follow us on Twitter, please go ahead and do so. There's stuff happening on that. And thank you for the opportunity, Thomas.
Thank you for the talk, extremely important work, and we'll come back to that in our discussion about standards for clinical assessment. You're a bit over time, nevertheless, if there are a number of questions, please answer very shortly. My question, can we use this in other languages? Can we simply translate it into German, French, and use it Portuguese?
I think the answer is yes. So this is a relatively straightforward translation. Yeah.
I think, yeah. Second question comes from Chip Wilmot. Would you think that the retest reliability is high enough to be an outcome in a clinical trial?
Yes, it's higher than that. The retest reliability was look at the ADL domain. Where is that score? Got this in.
Okay,
the
sure.
ADL domain is 0.9.
Your screen so that the next speaker can share his screen.
Sure. The ADL domain is 0.9, which is strong, and this is 0.86 for the physical domain. So I think we're higher than that. These are between mid 0.8s to 0.9. This is mental domain, which is different. I think this is fine.
Yeah. And then again, another short answer, please. Have you already some ideas about items which are specific for certain genotypes, or is this too early to answer this question?
So we have the data to look at that. We know the diagnoses of the people who did the retest reliability, but I think what we're planning, and we'll be starting soon with the CRC SCA, is a nationwide assessment of this going forwards. And I think there we will have both the long form and the short form. We'll have the genotypes, and we'll be able to figure that out, both with CRC and ReadyScar studies, to really get an answer to that question.
Okay, thank you. Thank you again, Jeremy. We can move on to the next talk that is given by...
Thomas, sorry, maybe just one housekeeping remark again. Just for the audience, for all attendees, don't be scared. We are just cleaning up the chat, the Q&A chat box, the Q&A box each time after each talk, and we're copying those questions and we're going to put them up back at the virtual coffee room. So your questions will not be lost. We are putting them back up to all the speakers in the virtual coffee room. So don't be scared if it comes cleaned off after each talk.
Okay, next talk, Adam Vogel. What is the time now in Melbourne, Adam?
It's near 2 o'clock in the morning.
Okay, so, heroic that you are participating in.
Thanks, Thomas.
Particular thanks to you to be here. And your talk is on speech as a biomarker in ataxia. What can it tell us and how should we use it? Please go ahead.
Well, thank you, everybody. Yes, it is 2 o'clock in the morning. I looked down the participant list. There's only one other person from the Antipodes. So Richard Roxburgh and I are up in the dark hours of the night. It will affect my ability to think, but just persist with me as I present through. So I'm pretty keen on speech, and people they often ask, why would we dedicate a significant amount of time to our protocol on a domain that we can assess relatively easily by a clinician's ear? Well, we know that speech changes as a function of disease progression and onset in the ataxias. It's an incredibly important aspect of a patient's profile because it dramatically affects, well, a disorder in it dramatically affects people's ability to maintain social relationships, keep a job, normal activities of daily living like asking for something. And from our point of view, we've been working quite hard to be able to measure these features objectively using some signal processing and data analysis methods. Our group have been working on using speech in a number of contexts, and a particular focus is using speech to measure a therapeutic effect. So using the processes that we've developed and others have developed to tell us whether or not a compound has been effective in a drug trial. That's one of our primary goals, but we can also look at it in behavioral treatments, something that I'll discuss in a bit later. Differential diagnosis is a tricky one. People like to think that they can make a diagnosis based on a large number of features that they may be able to derive from speech. I think the phenotypic descriptions from a speech point of view in the ataxias is fairly limited, but we can slowly work towards building a fairly clean phenotypic description of our different populations within the ataxias. It's lovely to see Louis on the call earlier. We've done some work in pre-symptomatic SCA2 and using speech as a subtle marker of performance in that regard. We've got a slide on that in a couple of minutes. And I guess one of the helpful aspects of speech in the experimental process is its application in different contexts outside of the clinic. So we can use speech perhaps as a home monitoring system, maybe not to describe the entire disease, but at least to describe the aspects of function that are relevant to communication. And not to harp on it, but we can measure speech in the context of an overall measure of speech-related quality of life. It gives us really tangible outcomes that reflect some aspects of overall disease severity, but importantly, speech in its own right. When we do describe speech, it can be both language and speech production. From an ataxia point of view, we focus on the motor manifestations of the disease, relating to respiration, voice function, how clearly we say sounds, so articulation, and also prosody. There's some growing interest in using speech, speech as through the domain of language, so looking at people's use of rules that govern, let's say, syntax, morphology, as well as semantics. And some work has been done by colleagues of mine in Melbourne looking at speech perception. So when we talk about communication, it can encapsulate all three domains in that regard. From a experimental protocol design point of view, there are a few aspects that need to be considered if we're going to use speech in a clinical trial setting. So protocols need to be designed to fit across the lifespan. So children do not provide the same samples as adults, and there's a complex period in our life called adolescence, which means there's a transition in how our speech production is manifests. It needs to be simple to administer, so both and complete. So from the participants and the testers' point of view, there has to be some simplicity there that mean that we can provide that same protocol over and over again. So that repeatability is incredibly important. Thomas has touched on whether or not we can have easy translations of protocols. It's not straightforward for speech, given that there are some differences between languages, but that's something that needs to be considered. And we've seen some growing interest in certainly pharma trials where they want a mix of clinic-based assessments and some home monitoring to give us perhaps a truer picture of function across those two experimental settings. I just wanted to, I've got a couple of quick data slides that I wanted to highlight. Mattis and I and team have done some work in building up a stronger phenotypic description of some of the rare ataxias. And here are two examples of studies that we've completed in the last few years. One on POLG, but another on RSACs, where we've got a couple of goals. One is to establish a baseline for the modal or habitual speech production in the population that we're investigating, and the data that we have here on the right is describing in an objective way differences between a particular population, RSACs, and age and sex match controls, where we can see there's no deviance in some of the tasks that we're eliciting, so voice quality, for example, but huge changes or differences for aspects of timing. This gives us information on the measures that may be suitable for later trials, and so follow this study up with some longitudinal data and then some sensitivity studies as well. Two approaches that we've been taking in other ataxia populations relate to both data use and also the potential sensitivity of it in pre-symptomatic stages. So the first study I want to describe is an approach where we've taken a relatively large sample of individuals with Friedreich's ataxia, and our goal in this setting is to derive some objective composite markers of two important ways of describing speech. One is intelligibility, so whether or not we can understand an individual, and one is naturalness, and whether or not they are a deviation from a healthy norm. And what we have done is applied a large number of acoustic features to our protocol. We've then asked two expert raters to go through every single one of those samples, blinded to stage of disease, but in the end they still know it's Friedreich's ataxia. And we've been seeking to develop a composite measure of those two perceptually derived expert-derived ratings of dysarthria. And this data is just about to be submitted. So one of these 180 people on this call likely to be reviewing it, hopefully positively. But what we found is that we're able to approximate those measures of intelligibility and naturalness using a composite feature of the acoustic measures with lower variability than the expert raters themselves. So MAE stands for mean adjusted error, and out of a 5-point scale in this setting, we had an error of around 0.3 for naturalness and 0.4 for intelligibility. Happy to describe them in any other way at another time. This study on the right is our examination of pre-symptomatic spinocerebellar ataxia type 2, which was just recently published in Neurology, excuse me, where we ran a relatively wide protocol on our individuals. So we've got 3 groups here. We've got pre-symptom, sorry, I can't see my, we've got healthy agents, sex match healthy controls, we've got pre-symptomatic individuals, and then we've got early stage individuals. So these people were within 3 years of diagnosis. And what we were able to do is separate out our pre-symptomatic and healthy controls using these motor challenge tasks. So these are syllable repetition tasks where we're really pushing the motor system, and that was exposing their deficits in production. What we didn't see are deficits in tasks like reading or saying the days of the week. It's important to consider how we assess speech objectively, but it's hard to ignore the elephant in the room, and that is the patient self-report. So we've been building over the last year and a half a tool called the Dysarthria Impact Score, which is currently sitting at 22 items covering 3 domains of function, emotion, and physical response to dysarthria. We're currently reducing those items to a smaller batch, and this tool is looking like it's going to be sitting around less than 10 items. We've been validating this on different populations, including Huntington's, Parkinson's, and ataxia, but we should have this data ready for you very soon. My final slide is an important one for us. I spend my life assessing people's speech, but it didn't feel right to continue that process without also trying to improve it. So over the last 9 years, we've been slowly building up evidence to support a biofeedback-driven dysarthria treatment, and I'm happy to say that we've just finally closed recruitment on a large multi-site, multi-language RCT on a treatment for Friedreich's ataxia, but also spinocerebellar ataxias. I think we're up to 117 individuals at close of recruitment. These data are undergoing analysis, and we should have it for you very, very soon. Thank you for your time.
Yeah, thank you, Adam. Currently, there are no questions, and unfortunately, we are running a bit out of time. So thank you again. Would suggest that we move on to the next talks. Both talks that are still in this session have a 10-minute slot. So Faye and also Winfried, your talking time should be 8 minutes or so. Next speaker is Faye Horak from Chicago on wearable sensors for multicenter clinical ataxia trials assessment and clinics lab. You're still muted, Faye.
She says she cannot share screens. Faye, it was working for the other ones. So you should be able to share your screen, Faye. Not sure it works. Maybe while working that out, maybe we can then switch. Winfried, are you ready to share your slides? Winfried?
I'm there. Yeah, probably.
So maybe I can just, while you're putting up your slides, just start sharing. Maybe I can just introduce Winfried Ilk. Winfried Ilk is doing movement analysis with a strong focus on degenerative ataxias. And while his talk is going to focus about the movement analysis in real life, Faye's talk will be more focused on lab-based movement analysis. So we have 2 complementary talks, and Winfried, it's your turn to talk about real-life gait assessment in real life.
Can you see my slides?
Yes.
Okay, and then thank you for the invitation and the introduction, and welcome to all to my talk, Real-Life Gait Assessment in Cerebellar Ataxia. You all know that with disease-modifying drugs on the horizon for ataxias, motor biomarkers are highly oriented. However, the evidence of ecological meaningful improvements requires the identification of patient movement behavior in real life of the patients. So far, the predominantly ataxic state is and its treatment-induced improvements has captured in lab-based assessment by us and by others. And the problem to transfer all this to real life is that movement variability is increased in the real life, also in healthy controls, and our ataxic measures predominantly are based on variability measures, variability of step length or step time. So the objective was to unravel specific biomarkers of ataxic walking and gait in real life. So we used therefore these same sensors as Horak. These are APDM inertial sensors. We used 3 sensors here at the feet and on the pelvis. We assessed 43 patients, and the measures were mainly spatial temporal step variability, harmonic ratios for measuring smoothness, lateral step deviation, and a compound measure of spatial variability combining lateral step deviation and step length variability. And we assessed in 3 conditions. In the normal lab-based condition, 15 meters straight walking in the clinic. And the second condition was this 5 minutes supervised free walking. And the third condition was capturing the movement behavior of the patients at patients' homes. And if we look at the results here for the first parameters, is step length variability, in blue healthy controls, in red the patients. You see in the lab assessment there's a high significant difference between the patients and the healthy controls, but in real life the variability of the healthy controls is increased, so the significance is getting a little bit weaker. But on the other hand, our parameters here, you see the lateral step deviation and the compound measure of spatial variability. And here you see also this high significant difference between health controls and patients in the real life with a rather high effect size. And coming to this sensitivity to ataxia severity on the left hand, first in the cross-sectional data, we subgroup the patient according to this HARA posture and gait score in mild affected, in moderate affected, and in severe affected. And you can see here by the parameter of right time variability in the lab assessment, there is a significant difference, but this is not anymore there for the real life. But for our compound measure, we have also a significant difference between the subgroups in real life. And currently, this is new data. We have done a 1 year follow-up assessment, and in this longitudinal data, we can show that there is a change in the lateral step deviation after 1 year. We have seen no significant change in the SARA score, had in this walking parameter in real life. And his height is great walking. I want to focus your attention in a more complex walking behavior. This is turning movement. The hypothesis is that turning movements may challenge the dynamic balance control more than walking, and so turning may reveal some changes in very early phases of disease. And for turning, we have looked on a measure which regarding the lateral acceleration in specific turning phases. And when we look in this, and in this study, we have also pre-ataxic mutation carriers in the population. Here you see that we can, here in green are health controls, in blue the pre-ataxic SCA mutation carriers, and red the manifested SCA patients. Here we can, in real life, we can distinguish also between healthy controls and pre-ataxic mutation carriers. And also, if we look for the sensitivity to ataxia, so severity, cross-sectional and longitudinal, you see a very good correlation between our LVC measure in all conditions, in the web-based condition, in this supervised free walking, and in the real life turning condition. And also here we were able to find in the 1 year follow-up assessment and significant change from the baseline to the 1 year follow-up assessment. So I come to the conclusion: while the assessment of constrained tasks at snapshots and visits represent patient real-world function only in a limited fashion, our measures of real-life motor performance can add ecological validity. And besides increased movement variability in real life, our measures can show a higher sensitivity to ataxia severity, with highest effect sizes actually in the real life assessment. And in particular, the turning movement could be very interesting for early stages, for pre-ataxic stages of ataxia. And in the future research, we want to complete this picture of real life movement behavior. But I think I can conclude that our measures are valuable candidates for both progression and treatment response outcome parameters in upcoming SCA trials. And so I want to end with all the people involved in this project, and thank you for your attention.
Okay, thank you, Winfried. One short question. Have you any data on stability of these measurements? So if you do these real-life measurements on 3 consecutive days, you have the same findings, or how much variability is there?
We haven't, and in fact, we have not concluded in this study, but it looks, we have this done for 10 or 11 patients now, and it looks pretty stable, but it's too early for really claims on that.
Okay, thank you. Then we move on to the final talk now of this session. I hope that Faye has solved the problems, appear to be MAC problems. But it should work now. I see your face, and Faye Horak, welcome, and you can start with your presentation on wearable sensors for multicenter clinical ataxia trials and assessments here in clinics lab.
Sorry about the technical problems. I think I need my coffee early this morning. So Thank you. As you'll see, I'm going to be also talking about using the same kind of wearable sensors as the previous speaker, and you'll find some of the same measures of gait that are sensitive to spinocerebellar ataxia. Whether in this case, it'll be for a 2-minute walk rather than walking at home during natural life. This is a collaborative study in which Dr. Gomez from University of Chicago collected most of the data, but there's many other people involved. So the purpose of the study is to identify a set of gait measures that are very sensitive and specific for spinocerebellar ataxia, both prodromal and those with manifest ataxia, and by comparing them to age-matched healthy control subjects, and then to determine how these top measures relate to disease severity. In our case, it's duration of disease and the SCA. This work has been submitted for publication recently. And I do have a conflict of interest in that the sensors, I helped develop the system that makes the sensors, and APDM, wearable technology that makes the sensors, was recently joined a company called ERT that has technology in over 75% of clinical trials. So the sensors are these inertial measurement units or movement monitors called OPOs, and subjects wore them on their belt, on their sternum, on their wrists, and on their feet. And we had quite a few patients with ataxia. We had 163 people with spinocerebellar ataxia and 96 healthy controls of about the same age, about 50 years old. And you can see that we had SCA 1s, SCA 2s, primarily from Cuba, SCA 3 and SCA 6s. And for the prodromal subjects, those who had SARA scores of 2 or less, we had 42, and we also compared them with the controls because they were about the same age. And the first way we looked at separating, identifying which measures of gait were most sensitive to SCA. We used the area under the curve for the ROC curves, and all these measures are rated from the top with the most sensitive measure, which I repeated here on the left, and to the least discriminative measures. And you'll see in red are variability measures of gait during the 2-minute of natural pace walking. And so variability, even more than the means of gait, were very sensitive to SCA. And for example, the toe out angle variability or standard deviation had an area under the curve of 0.94, which is almost perfect in separating those with and without ataxia. And double support times variability was another one. And toe out angle is an important one, as I'll be showing you, because it relates to the stride width, which we know is naturally too wide and too variable in people with ataxia. Double support time is important for balance. Other measures that were sensitive and specific were toe off angle, as the previous speaker had noticed, and variability of that, and elevation of the foot at mid-swing, and also trunk stability. Now, I just wanted to mention something about our most sensitive and specific measure, which was toe out angle or external rotation of the foot. There's some studies in the biomechanics literature by our Quo's laboratory showing that you keep your balance by not only widening your stance when you walk, but by externally rotating a foot. That kind of steers you as if you're riding a bicycle and helps you keep your balance. So the early measures of balance problems we think can be reflected in how the feet are used to control balance and also stability of the trunk while walking. As you see below here, stance width in his study was correlated with rotation of the foot. The other thing we did then is take the top measures, those that were most sensitive and specific, and to look at how they were correlated with disease severity and duration of disease. So we have toe out angle, which is that stride width variability, and double support time variability down below. And you can see they're highly correlated with disease severity as rated by the SARA score, and also with disease duration. These are, so both the most discriminative measures were both sensitive to disease severity and duration. We wondered whether it mattered what kind of SCA you had. So we looked at comparing the control values for our top measures with each type of SCA, 1, 2, 3, and 6. And here in these box and whisker plots, you see that all types of these 4 types of SCA were sensitive to toe out angle variability and double support time variability, toe off angle variability, and elevation at mid-swing variability. So those are 4 different measures. The first one related to stride width. The second one about double support is related to balance problems. Toe off angle variability, I believe, is related to coordination, and elevation of mid-swing to hypermetria or high stepping. We looked at these 4 measures then and to see how they correlated with the SARA score for each different kind of SCA. And you can see there's good correlation across all the 4 SCAs for all these 4 measures that are discriminative. However, when we looked at disease duration, it was primarily SCA1 and SCA6 that showed strong correlation with duration of disease, whereas SCA2 and 3 were less so. They were not as sensitive to disease duration. We also had 42 people with prodromal SCA, that is those who on a SARA scored 0, 1, or 2. And we found that the same measures, in red here, the variability measures, were the most sensitive measures in separating them from healthy control walking. But of course, it's not a 0.95 anymore. You know, our area under the curves are like 0.67. But some of the same measures like toe out angle variability was very sensitive. The other ones that were more sensitive in prodromals than later were what the trunk was doing, the sagittal motion of the trunk, the lumbar rotation of the trunk, and things like that. But they all are standard deviation measures. We didn't have any longitudinal data right here that we measured, so we compared the most discriminative measures in our prodromal SCAs with the average measures in our manifest SCAs. And you can see the slopes here being pretty similar. Of course, these are different people. And in our next study, we'll do a longitudinal analysis in the same people to see if it holds up, where our top 5 measures of variability of gait, are they also very sensitive to progression in longitudinally. So in conclusion, we believe that wearable sensors like these are useful for clinical trials and clinical practice. That gait variability is the most sensitive and specific characteristic, and we saw that for home monitoring and as well as for this 2-minute walk. And we believe that eventually we'll be combining these measures along with sway measures that can be quantified during quiet standing with a single sensor on the belt, and using that as a combined mobility measure for future, as a biomarker, as a performance biomarker to monitor disease progression for FDA submission. Our natural history study is going on now, and I won't tell you too much about that, but it's a cooperative study among several sites to look to see whether both balance and gait measures are going to be appropriate for clinical trial outcomes. Thank you.
Okay, thank you, Faye, for your talk. There's one question coming from Bart van der Warrenburg in Nijmegen. Did you explore performance of some single sensor measurements, as this would be of benefit for wider implementation?
Right. So all the data that I talked about here just comes from sensors on the feet. And these sensors provide more accurate information about what the feet are doing. If you had a single sensor on the belt to look at gait, for example, you would not get toe out angle or elevation of the foot, or a lot of these things that we found that were most variable. You could estimate some aspects of gait that would be more noisy. So your effect size wouldn't be as good because you wouldn't have as accurate measurement of what the feet are doing from a single sensor on the belt, for example.